Welcome back to Principles of Organismal Biology. We are initiating the second week. That's exciting. And today we are going to cover the evolution of the genome. Do you remember what a genome is? We have covered it. Think. Okay, so genome is all of the genetic information that an organism has. And this is mainly DNA, except on virus. Some virus can have the genome made out of RNA. So the DNA is in these cells. We have seen the locations in some cells, and today we're going to see how they evolve. I hope that you like this class. So the main objectives today is to learn what genome evolution is. We're going to see the importance of bioinformatics and how it impacts the study of genomes, and we're going to compare the genomes between prokaryotes and eukaryotes and identify the main differences among these two lineages. But first, how do genomes evolve? You know this answer, and that's through mutations. Last class, we learned that mutations are responsible for the genetic variation, and it's very important when this mutation happens in the gametes, because that can pass to the next generation, whereas if it happens in the somatic cells, it doesn't pass to the next generation. We also learned that there are two types of mutations. The first one, the small scale, and it's usually associated to point mutations. It's a single nucleotide base pairs or a few associated to deletions and insertions of pieces of DNA. The other category is large-scale mutations, and those involve large arrangements of the chromosomes. This is an example of what happens at the chromosomal level when there is a duplication or inversion, and it happens during the meiotic recombination. In this case, we have two chromosomes, one coming from, let's say, your mom, and another one right here coming from, let's say, your dad. So these chromatids are aligning, and if they are properly aligned, they are gonna exchange some pieces of DNA. That's part of the recombination that happens in prophase, in meiosis, prophase 1, and we call it also crossing over. This is the crossing over point right here. However, what happens when they are misaligned? So in this case, you see how we have a similar sequence. So this sequence right here in yellow, it's called transposable elements. And transposable elements can be, they're also called jumping genes, and you can have the same sequence of the same transposable elements in different parts of your chromosome or in your genome. And then therefore, the chromosomes can be confused and align in these areas, but they are not properly aligned, as you can see here. As a result, when the crossing over happens, they have this crossover point in this transposable element, for example, it's going to result in a chromosome or a chromatid that has a longer arm and another one that has a shorter arm. And therefore, here we have a result of genome evolution. So for you to understand a little bit more about what transposons are, I have added this figure in which you have the transposable element and some inverted repeats. This transposable element is going to be inserted in this target. So every time that in your genome you find this sequence ATGCA, this transposable element can target that area and copy and paste itself there. And then you can just have more copies of this transposable element. If you think about it, that also leads to genome evolution, because the copy of this transposable element is going to increase the size of the genome. We have gone through this example in humans, and I'm putting it here for you to review these concepts. So in humans, we have this chromosome number 2, and we have said that that's the result of the fusion of two chromosomes coming from an ancestor lineage, And we know that because in chimpanzees, we have chromosome 12 and 13 that share some sequences and some features to the chromosome number 2 in humans. So now genome evolution, we're going to be talking about these in the terms of the comparison of different genomes with respect to each other and see how they have changed through time in different lineages. This has been done thanks to the massive amount of sequences that we have generated in the last 2 decades. That's a very important discipline that we have extensively, or that we are studying extensively. In my lab, we actually do some of these genome studies. And genomes can vary in size, in the number of genes, or in the gene density. Here you can see a table with all of the information, not all of the genomes. These are just a subset of genomes, but you can see how they are sorted. So they are sorted by size. And at the top, we have the smallest genome, and at the bottom, one of the largest genomes. And at the top, you can see that bacteria, Haemophilus influenza, for example, has one of the smallest genomes. E. coli, the one in the blender experiment that we covered already, is also very small. These 2 are examples of bacteria. We also have archaea here with the small genomes. And bacteria and archaea are part of this artificial group called prokaryotes, which is a group that lacks nucleus. And eukaryotes are all of these species that have a true nucleus, and they start to have larger genomes compared to prokaryotes. So in eukaryotes, we have a very wide range of genome sizes. And let's find our results. We are right here with 3,000 million base pairs. 1 million base pair is equal to 1 million base pair. A mega base pair, it's equal to 1 million base pairs. So this is 3,000 million base pairs. So eukaryotic genomes tend to be larger, and most plants and animals have genomes that are larger than 100 megabase pairs. But let's see, what's the plant that has the largest genome? There are several, but one of the largest is actually a pine. So I guess before we have the sequence of the genome, the human genome, and all of the other genome sequence after, we thought that humans should have the largest genome because we are very smart, we have created these civilizations, but actually our genome is not that big compared to other species. Pinus lambertiana, this pine, has a genome that is 31 gigabase pair. Previously, we were talking about megabase pairs. In humans, we have 3,000 megabase pairs. This is an order of magnitude larger, and this is a 31 gigabase pairs. This is gigantic, the information that we have in this species and in other pines, but also in other plants. Now let's see, does that matter how many base pairs or the size of your genome and the number of genes that you have there? This graph right here explains that. And if you look at your x-axis, you can see the genome size. And in the y-axis, we have the number of genes. So let's take a look to this one. So in 4 megabase pairs, right, in 4 million base pairs, we will have 4,000 genes. If you do a quick math, you will figure out that that means that one gene, in average, one gene has 1,000 base pairs. 1,000 base pairs. And that's very, very similar for bacteria, shown here in blue, and in archaea, shown here in orange. So these two lineages of prokaryotes have, in average, one gene per 1,000 base pairs. So based on this information, if there are around 1,000 base pairs per gene, and the human genome is 3,000 megabase pairs, how many genes do humans have? This is a tricky question. Okay, let's see the answer. So actually, you probably picked A, if your math is right, and based on the information that I gave you. However, the right answer is 22,000 genes. How come? This is not matching what I just told you. But do you remember I just taught you about the transposons? And I told you that these pieces of sequences, or these jumping genes, copy themselves and paste to each other, to other locations, and increase the size of the genome. So this is what happens. So we have a lot of information that we don't understand. Some of it's part of these transposable elements right here, repetitive sequence. It's 44% of the human genome. It's repetitive sequences, and they are also pseudogenes, and pseudogenes are copies of genes that lose their function. We also have introns. Hopefully you remember these introns when we explain transcription. So we say that the mRNA, we have a pre-messenger RNA, and it has to have some editing in order to have the mature RNA. And in that editing, we are going to have pieces of DNA that get removed, and then the mature RNA is usually shorter. So those introns cover 20%, and they are not part of the codifying information. In other words, that information doesn't get translated into proteins or polypeptides. And we have other type of information that is not non-coding, around 15%, and other repetitive DNA that is not related to transposable elements, that covers the 14%. Basically, we only have 1.5% in exons. And the exons are the ones that exit the nucleus in the form of RNA messenger, the mature form, and they are the only ones that are going to get translated into polypeptides. So it's a very small percentage. Most of the other percentage is transposable elements. Cool, huh? Okay, now let's jump into bioinformatics. And this is something that my students in my lab do a lot, mainly every day. Bioinformatics is going to be a set of tools that are going to allow us to investigate the sequences in these different species and compare them. And we want to do that to understand the evolution and biological processes in these species. Now, we can compare different regions of the genome, and some of these regions are going to be very conserved. Now, think about this. What genes do you think will be conserved? I'll give you a hint. So think about processes that are shared among all of the individuals, among all of the species. So you can think about replication, DNA polymerase. So the information that codes for the DNA polymerase must be very conserved. Another one is the formation of the ribosome. The ribosome needs to be active all the time. It's a feature that is shared among species. And yes, the ribosomal information at the DNA level, it's very conserved. And there are other areas of the genome that are less conserved, and that allows us to investigate what makes a species, or in the case of humans, what makes a human, and separates them or separate us from the other species. For this, we have to go through high throughput sequencing. And these technologies have evolved in the past 2 decades. Before that, we used to sequence, but that's called Sanger sequencing, and it's not high throughput. We used to sequence a few hundred base pairs with that technology. We could sequence up to maybe 900 base pairs, and we were just starting to have troubles. With these new technologies, we can sequence full genomes. Some of the technologies are nanopore, Illumina, and pyrosequencing, although pyrosequencing is not being used that often. And right now, Illumina is one of the technologies that we are using heavily. That process involves sharing our DNA of importance or of interest. And this DNA will look like that, right? This is a representation, a graphic representation of that. And we cut it in multiple pieces. We could use enzymes that cut areas of the genome, or we could just share it by using ultrasound. And when we do that, after we do that, we can ligate some adapters. And those adapters are used to sequence, and we can sequence all of these different pieces and then assemble it. So all of these parts up to here involves lab experience, hands-on with pipettes, taking some tubes and enzymes and doing our libraries. But then once that we put it in the sequencer and the sequencer gives us actual sequences, that means the nucleotide sequences A, T, 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 G, T, 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 C, etc. Whatever we get, we need to make sense out of it. And we make sense with this bioinformatics. So that means the application of computational methods to store and analyze the data. And sometimes if the genome is really large, for example, in the pine that I told you, we need multiple clusters or several computers to be able to handle this massive amount of data. So it's very cool. If you like computers, you should be thinking about exploring this. And if you like the lab, this is also a very, very important tool that you want to learn. Also, if you like engineering, it has been a very nice relationship between engineers and molecular biology in order to develop these technologies. So this is also something to consider if you are into engineering. So let's take a look to this. This is Bioinformatics in Action. I want to share with you this webpage is the NCBI. And you could take a look to this because in this webpage, you could search for a gene and you can have a gene. You could also search for a genome and you could download the sequence of the genome of different species. And this is publicly available. That means that you, your mom, your siblings can have access to this information. Everybody can have access. I want to share with you this sequence. I want to show you this sequence of Thermus aquaticus. Thermus aquaticus is an organism, a prokaryote, that lives in higher temperatures. And usually its optimal temperature is around 70 degrees Celsius. And we have extracted the DNA polymerase of these species to use it in the lab. So if you are familiar with PCR, polymerase chain reaction, that enzyme that we extract from these Thermus aquaticus can be used in the lab to amplify a piece of a gene. And the trick is that this is one of several species of a microorganism that can handle these higher temperatures. So we can modify these temperatures in the lab in a machine that changes the temperature from, let's say, 55 degrees to 72 to 94 in order to separate the double-stranded DNA, amplify the region, and at the maximum temperature, at the optimal temperature that this enzyme works, which is around 72, 70. Okay, let's take a look at this. Okay, in this case, we're going to be typing NCBI here, and that will take us to the National Center for Biotechnology and Information. If we click there, we could look at all of the databases, and we're looking for Thermus aquaticus. So if we click on this, it's going to give us all of the information located in the NCBI. As you can see here, there is a genome sequence for this species. So we have all of the sequence in this genome. But we also have a lot of pieces of this genome in this axis and this link, and they are the nucleotides, so pieces of genes. And if we are curious about this Thermus aquaticus, the DNA polymerase 3, that is going to give us the information. And in here, we have the sequence of the polypeptide, right? The M, it's the methionine, glutamine, serine, etc. And here at the bottom, we have the actual sequence of the DNA of this gene before getting translated. So GCC, GAA, etc. And we could see that this gene is more than 4,500 nucleotides. We could also look at the protein, and this is very cool because we could see the structure of the protein in 3D. So all of this is for Thermus aquaticus, different models of the protein, and let's click on this and see what it says. So in here we have different colors representing different subunits, and we could click on this or this. Either will give us the 3D, and this is just a 2-dimension representation of this protein. We could see all of the subunits and in here. So here we could zoom in and out, but we could also rotate this molecule and look carefully. Hopefully you can see here, or at least this seems familiar to you. This is the DNA. So if you remember, I told you that this is the DNA polymerase, which is helping with replication of the DNA. So it's copying this DNA, and obviously this DNA strand is really, really long, or it could be very long, but this is just a model for you to understand. Okay, so I hope that you have enjoyed this section. You could look for anything in these databases. So please just think about your favorite enzyme, maybe your favorite gene, your favorite pet, your favorite species, and search in the NCBI as for a fun homework for you if you want to play with it. Okay, now let's just talk about the Genome project. I have mentioned that the first draft was published in 2001, but the actual project started in 1990, and the final published version of the first genome was completed in 2007. So now we have a lot of genomes since then. A lot of people have sequenced different humans, and so we have many genomes of the human species. But we also have sequences of other genomes. So despite of having a lot of sequences of different genomes in the human species, we are still trying to investigate what are the functions of particular genes. And once that we figure it out, that's going to be very important in medicine to identify genetic diseases and, for example, prevent expression of some lethal genes or deleterious genes that we have learned during the mutation when we talk about mutations. So it's very important to have this discipline moving forward. As I quickly mentioned earlier, really understanding or sequencing the genome and comparing it against some relatives, for example, comparing humans and chimps, will allow us to understand what makes a mammal or what makes a human and what genes or what genetic information we have in different to other species. This is an example of the FOXP gene, and this shows rapid or shows evidence of rapid change in humans. This gene has been mutated in some animals, for example, in mice. And in mice, the mutation of FOXP2 leads to malformed brains, and also these mice over here are unable to emit normal ultrasonic vocalization. So it seems that this mutation affects some vocalization. Mutations of FOXP2 in humans affects the language and speech impairment. It seems that this gene, FOXP2, is needed for vocalization in mammals. It's also important that humans and chimpanzees seem to differ in the expression of FOXP2. And if you follow the news, you probably are aware that the sequence of the genome of the Neanderthals, where it was sequenced. Okay, if you follow the news, you probably know that the genome of the Neanderthal was sequenced recently. And when we look at this genome, we find that FOXP2 is identical in Neanderthals and in humans. And if you think about it, that raises the question whether Neanderthals could be able to speak. And also if it could communicate verbally with humans, but that's different because that involves language. But it's very, very interesting, I think. Now we can compare genomes, genome sequences, and it will provide us the evolution or clues to the evolution of certain lineage. In this case, I picked chlamydia phages. So chlamydia probably you're familiar with because you don't want to catch chlamydia. Chlamydia is a bacteria that will can affect humans. It could affect cows, for example, and they can get blind with that. But in human is mainly associated to, is considered a sexually transmitted disease. And which is interesting is that there are some phages, some virus that can kill the bacteria. Similarly, as we were talking about these phages when we talk about the blending experiment, yeah, those are similar bacteria, but similar virus, but in affecting different bacteria. This is an alignment or a comparison of different chlamydia phages. And you can see how these regions change in length because there are more base pairs in, let's say, this species of group 3 or this variant compared to this other one. And some parts of the genome also can change positions. So for example, the red area right here, it's towards the end, whereas in this species, it's after the blue region. So we need to compare full genomes to understand what has happened through the evolution. And this gets more complicated when we compare large genomes. So virus have some of the smaller, or have the smaller genomes, and later we have the bacteria and archaea, and then the largest will be eukaryotes. Which sequences are more closely related? What I will say is that the sequence or allele 1 and allele 2 are more closely related because they are only one base pair apart, whereas any other combination will be more than 2 base pairs apart. Okay, so for example, if you compare this against this one, this has mutated in 1 and 2 positions, whereas this only has one mutation. We could say that these two are most closely related, and we can make this diagram. In turn, these two sequences are more closely related to this other one than to this guy over here, because this is 3 base pairs different respect to each of these, whereas this one is only 2 base pair difference. And then this is the most dissimilar. Which sequences are more closely related? So here we have 3 alleles of the same gene, right? the same locus. So hopefully you pick these 2, allele 4 and allele 4. These are identical. This is like twin sequences. So something that you will expect in some twins or in very closely related individuals. Now we have these other variants, these other alleles. How closely related are they with respect to these other 2? So these are more closely related later, and then we have these this relationship with the sequences. Hopefully you start, this is more like building a puzzle of what's more closely related to each other. And this is what we do for fun in our lab. I hope that you have enjoyed this class and that you are starting to get excited about genome evolution. I'll see you next time.